Discussing C. elegans touch response. Here it's a new project that I started quite recently after moving to UCSD. And in particular, what I'll be discussing are two properties of this touch response that I got particularly intrigued by, which are adaptation on the one hand and the symmetry that I will define momentarily what it is, symmetry. It will be between onset and offset of the response. Most of the what I will tell you today is mostly contained in this paper that will come out or has come out this week in PNAS. And it's been done with Miriam Goodman. She's at the Department of Cellular and Molecular Physiology at Stanford, and Beth Pruitt. She's at Stanford as well in the Department of Mechanical Engineering. So let me get started with just by giving you a glimpse of the diversity of the receptors and the sense of touch of us, human. And there's, as usual with humans, there's an amazing diversity of receptors that we have. And here you have a list. For more details, it's going to be a sketchy on humans, so it's this nice review which has been written. This is just a part of the receptors that we host. And there's two broad classes, two broad distinctions that are made for receptors. First of all, it's whether they adapt or not adapt. Adaptation is defined as Adrienne was defining this morning. You put a constant stimulus and you look whether or not the nerve and the receptor stops responding. If it stops responding, it's classified as it's classified here as R, stands for rapid adaptation. So there's a class of receptors which is rapidly adapting, and there's other receptors that are slowly adapting, like here. The other distinction which is made is on the threshold. There's some receptors that respond to very strong mechanical stimuli, and others that respond with very low threshold. So that's the other distinction which is made here, low threshold mechanoreceptors, and then you have high threshold mechanoreceptors as well. Now, most of what I'll be discussing, the closest receptors, the closest to those that I'll be discussing in C. elegans, are rapidly adapting and low threshold. And among them, the two of them that are the closest, and as you will see, most relevant for what I will discuss, are the so-called Meissner corpuscles and the Pacini corpuscles. So they are, both of them are located in glabrous skin. Meissner corpuscles are located closer to the surface of the epidermis, to the skin. And Pacini corpuscles have this very characteristic shape that you see here. They're located deeper into the skin. And this affects the receptive fields, which is broader for Pacini corpuscles than Meissner corpuscles. Now, both of them, they rapidly adapt, and rapidly it means that they adapt on time scales that are of the order of 10 milliseconds. That's the time scale of adaptation, as you will see, and this is analogous to C. elegans receptors. And the other thing is that they function as bandpass filters. So they don't respond to very low frequencies, they don't respond to very high frequencies, and they tend to have a peak in the middle. Meissner receptors, they have a peak at about 10-20 hertz, in the order of the 10 hertz. And Pacini corpuscles, they have peaks in the order of a few hundred hertz. So you just imagine a usual bandpass filter. This has been characterized in both of them. Now, as usual with human, it's a little bit complicated. We are interested, of course, in human, but it's complicated to do genetics, it's complicated to do, to dissect, it's complicated to do molecular biology. So people have interest in model organisms, and as usual, a very convenient and very useful model organism is C. elegans. Now, in C. elegans, the usual reasons for having C. elegans are for touch, as for the others, molecular biology tools. It's the fast reproduction time, it's the fast that all cells are characterized, all the genealogy, and you can look through the organism. There's one more thing which is characteristic of touch, and it's the fact that C. elegans has a very well characterized behavior which is associated with touch. And this behavior is this escape behavior that I'm showing you here. So if you touch C. elegans in the anterior part, it will start moving and escaping backward. If you touch C. elegans in the posterior part of the body, it will start moving forward. And if C. elegans is moving forward and you touch in the back, it will accelerate and speed up. So this is an escape reaction of C. elegans, which is extremely easy to assay. It's extremely easy to characterize. So it's very useful in addition to all the other nice properties of C. elegans that are generic for molecular biology. C. elegans is also very useful for this reason. And molecular biologists have characterized this behavior. By the way, getting back to adaptation and habituation, I'll be talking about adaptation. There's another phenomenon, which is the phenomenon of habituation, which is that if you keep tapping on C. elegans, let's say you keep tapping with the spacing of 1 second, and you keep doing this for a minute. After a minute, C. elegans will stop having these escape reactions. This is habituation. It's not adaptation. The neurons will still fire. There will still be a response of the neurons, but just the brain will not process this information anymore. So the adaptation that I'll be talking about is on scales of tens of milliseconds. And then there's another phenomenon, which is habituation, which has to do over minutes, and it's not something that I'll be discussing. Okay? So people using this, let me show this again. And using this, behavioral assay, they've been able to characterize the the mechanosensitive neurons in the worm, and in particular to characterize six touch receptor neurons. TRN stands for touch receptor neurons, which are shown here. I've shown 4 of them that are here. This is a sketch of the body of C. elegans. So, as you know, C. elegans is about 1 millimeter long. The radius, the diameter of the worm, it's about 50 microns. And there's 2. You can consider this the inner tube where you have the intestine and the pharynx, and then you have another outer layer where you have the epidermis, which is sustained by the muscles. There's an outer layer of epidermis, and then you have these touch receptor neurons. I don't know if it's quite visible, but here there's a purple line which is running along the body here, which corresponds to a touch receptor neurons. So you have these touch receptor neurons that are running all along the body of the worm, and there's 6 of them. So let's pick this one, AVM. AVM stands for anterior, ventral, and then M stands for mitochondrial cell. So anterior and ventral, it's quite clear what it is. M, mitochondrial cell, the reason they're called mitochondrial cells is that when people start looking at them, the most striking feature that they saw is this bundle of mitochondria that you see within the neuron, within the cell. And therefore, they call them mitochondrial cell. They play a role, even though they are not the sensitive part, they're not the sensitive part of the neuron, but that's something that they saw, and that's why they call them mitochondrial cells. So that's AVM. And then you have ALM, which is anterior lateral mitochondrial cell, and there's 2 of them. There's 1 on the left and 1 on the right, so that makes for 3, and another 3 are located in the posterior part of the worm. And these are the touch receptor neurons that are responsible for the escape reactions that you've seen before. And the typical forces that are exerted in this kind of behavior are on the order of 10 micronewton. If you go to 100 micronewton, there's another class of receptors that are getting involved. And then there's another class, yet another class, that are located close to the nose of the worm that are mostly involved in foraging. For a total, I think, hermaphrodite, they have 30 mechanoreceptor neurons. So we'll be looking at these 6 here, and And if you want to know more, there's Miriam, she wrote in the chapter on mechanosensation in the worm book, so you can have all the details. Now, when you look at the, so now you can do mutants. You can do mutants, and you can start chasing the molecules that are involved in the response of these neurons. And there's a number of them, but there's one which is particularly important, which is the channel itself, which is called MEC4. It's a channel, it's a sodium channel that is amiloride sensitive. And you can see this has been stained here along the neural tube. So you have this neurite, which is running along the body. And then you have these channels, these MEC4 channels. This has been measured in this paper in 2007. So you can look, you can stain these MEC4 channels, and they have a broad distribution of spacing along the neurite. And they're spaced by a mean distance which is of the order of a couple of microns. So you should imagine this neurite, this tube, which is running along the body of the worm, and every couple, with broad distribution, every couple of microns, there's a channel which is this mechanosensitive channel, which is MEC4. Of course, in addition to the channel, which is here, you have a bunch of protein, structure, protein, collagen, and other things that are positioning the channel here, and they are providing the structure of the channel itself. But the main object is this sodium channel that you have. So now the question is, how is this channel gated? And in this, having C. elegans is particularly useful. And in particular, it's possible to do this kind of experiments here, where you have a cantilever, piezoresistive, which is controlling the movement of a ball, which in our case is of the order of 10 microns. So controlling with this cantilever, now you can deliver mechanical stimuli on the body of the worm that you have here. And in terms of mechanics, it's very useful, and there's many biophysics experiments that have been done, and it's not too bad, it's not too wrong to think of the worm mechanically as a shell. Here, the thickness of this shell is of the order of a few microns. And then there's the internal part, the muscles, the intestine, and the inner tube that are providing some pressure, which in fact is of the order of about 40 kilopascal in the intact worm. So you can think of this as a pressurized shell, which is maintained by the internal pressure. And so you stimulate mechanically with this ball, which is controlled by a feedback system. And the other thing, which was the main step forward in the field, was that they were able to patch clamp on living worms and have, as you can see here, have a recording pipette. You expose with a razor. What you do is that you cut the body of the worm, you expose the neuron that you're interested in, you go there with the recording pipette, as you can see here, and then here you have the cantilever, which is delivering the stimuli. So you have a very well-controlled system that delivers mechanical stimuli, and then you pick the response of the neuron, which is the current which is flowing through the neuron. So you have the input, and you have the well-controlled input and the output. Now, one thing that you can do with the worms that certainly you could not do with humans is to do the following thing, is to, in the worm, as I just said, the preparation is that you just expose the neuron, but you keep everything else intact. One thing that you can do is that you essentially change the pressure, the internal pressure of this tube, by taking the gonads and the intestine out. Okay? So you clearly change the mechanical properties of the worm, and now you can ask, how is the response of the neurons? How is it going to get affected by this change in the mechanical properties? And no surprise, the intact animals are stiffer than the animals where you took the intestine out. So the colors, they refer to this. The red is the intact animal, and the blue is the animal where you dissected the intestines. And clearly this is force versus indentation, so they measure how much you penetrate with the ball in the body of the worm. And clearly you can penetrate more into the animals where you dissected the intestine out. The pressure has been reduced, therefore you penetrate more than in the animals that are intact. And so now you can go and look at the response, and here is what you get. So this is the normalized current. So you normalize the current to the maximum value, and as a function of the force that you exert. And this is the blue. These are the soft animals, and these are the animals that the normal type. Clearly, as a function of the force, these two curves have nothing to do with each other. While if you look at the current as a function of the indentation, they collapse within the limits. All these animals are different animals, so there's a certain diversity among the animals. So I would say that these curves are certainly closer to each other than these two, which allows to conclude that the mechanotransduction current, they depend certainly more on indentation. The control parameter is the indentation of the ball within the body of the worm rather than the force that you deliver. So you have a, now because of this, Miriam and Beth, they constructed a system where in addition to having a force clamp system, they also have an indentation clamp system. So they can deliver either a force which is controlled or an indentation which is controlled.
What happens to the muscles in the animal?
The muscles are left intact.
And they doesn't twitch or doesn't, what is the response? I mean, there's this escape response.
Yes.
So it doesn't try to...
Oh, the animals are glued.
Oh, I see.
The animals are glued. Oh, of course they would escape. For sure. No, the animals are glued. So when they do the experiments, the animals are glued in the, where is it? Alciolator. So they're positioned like this. There's a glue which is keeping all the ventral part of the worm. It's kept fixed by glue. Yeah. No, they any other? No. Okay. So this is, by the way, I'm giving you a short version of the talk that Miriam Goodman, she came for a seminar at UCSD, and she told all this story with many more details, with all the mutants and all the assays and the other things, which is interesting molecular biology. But what really caught my attention is the following thing. So in 2005, together with O'Hagan and Marty Chalfie, they managed to develop this patch clamp system and to measure in vivo the response of the neuron. And what they saw is the following thing, is you deliver a step. So you indent with the ball, and then you keep it for a few seconds, and then you take it out. This is the voltage that they measure in the neuron. This is the current, okay? So when you apply the step, you get this strong current which gets to flow. On a time scale of the order of about 10, 15, 20 milliseconds, that depends on the animal, but on a time scale of the order of 10, 20 milliseconds, the response fades away and there's perfect adaptation. You take the stimulus out, you get another spike, and you get perfect adaptation. Okay. So the system, first of all, it's adapting, and second thing, has a symmetric response at the onset of the force and the offset of the force. And for reasons that I'll describe in a second, I was extremely surprised by the symmetry of the response for reasons that I'll describe momentarily. Before I get there, let me just mention that this property doesn't depend on the mechanical properties of the worm. If you do it in stiff animals or soft animals, you get the same symmetry. Once you accumulate statistics over different animals, you see that the onset and the offset response is statistically indistinguishable. Now, first question is adaptation. So the first question you have is adaptation mechanical or adaptation is chemical? So that's the question I asked. And adaptation, all the evidence is that adaptation is not chemical. The argument which is given is that the adaptation time of the order of 10 milliseconds, it's not consistent with chemical modifications of the time that we are familiar with in chemotaxis like methylation. This is not something that is consistent with those times. On the other hand, in neurons, it is known that calcium can give adaptation. The flux of ions can give response times of the order of 10 milliseconds or so. But calcium was taken out of these neurons and adaptation is still there. So all the evidence is in favor of mechanical adaptation rather than chemical adaptation. Now, mechanical adaptation is not a big, is not by itself, is not a major surprise. We have examples. I mean, visiting Stan, Albert, and Eric at Rockefeller, I was familiar with the example of hair cells. So let me just remind you the hair cells, what's the model. And you can get all the details in this review. The model of Hudspeth was proposed 30 years ago, but this recent review that makes a very nice review of the state of the art. So our ear is shown in a schematic way here. This is the external part of the ear that you have here. Sound is coming from here. And it impinges on the eardrum that vibrates and sends sound waves into the internal part of the ear. Now, these waves, they travel along the basilar membrane, and you can get all the details in this review of how this basilar membrane is tuned and is arranged in such a way as to maintain this wave and at the same time have dispersive properties that select the frequencies. It's an amazingly beautiful system. But for what I want to tell you today, the important thing is to look at these hair bundle cells that you have here. These hair bundles are in contact with the tectorial membrane that you have up here. And so when there's relative movement between these two parts, this is what a hair bundle looks like. They are in contact here, and they get mechanically stimulated when there's some sound. In particular, this one is called kinociliums, which is the highest one that you have here. And the way we detect sound is that these hair cells get mechanically excited, and they start responding. And in particular, the scheme and the model which was proposed by Hudspeth is the following one. You have the hair bundle with the kinocilium on this side and the stereocilium on this other side. And these different hair cells, they are connected by the so-called tip link. Now, the nature of this tip link is still a matter of discussion 30 years after the proposition of the model. So I'm giving you the 101 version of the model. The molecular nature, as I said, of these elements is still being discussed. But the basic form of the model is the following one, is that on these hair cells, you have the channels, which are shown here, and then you have a gate, which you should imagine is like a trapdoor, which is sitting on the channel, and it's gating the channel, it's keeping the channel closed. And then you have a filament, this tip link, which is here, and which is attached by an insertional plate to the next one, to the next hair cell that you have here. And then there's some molecular motors that are responsible for the movement of this up and down. So now let's look at what happens when you have a positive stimulation, which means you have these cells, they get the force that tilts the kinocilium in this direction here. What's going to happen, as you can see from this plot here, is that the tip links are getting stretched. So this elastic filament is getting stretched, is getting under tension, and the tension, what it's going to do is that it's going to raise this trapdoor that you have here. The ions will flow into the channel, and you have the current and the response of the neurons. Once the tip link is under tension, then these motors will move down here and will relax the tension in the tip link, and this trapdoor is going to close. So you can have mechanical adaptation, as you've seen here. This is a well-known model. So by itself, having mechanical adaptation is not the biggest surprise of the world. The problem is that if you think in terms of this model here, you get a problem with the symmetry. And that's what puzzled me, and that's what I was just, I couldn't make sense of the plot of the symmetry that you've shown before. Because let's look now at what happens if I apply forces in two different directions. So let's suppose I apply the force in this direction here. This is the stimulus. I'm pushing this way. So the hair bundle will start moving, and then it will stop, as you can see here. It will reach a certain position which is tilted this way. And this is what the current looks like. So as the onset of the force, you have a strong current. Then you have this phenomenon of adaptation, so it relaxes back. And then actually these motors, they have a little bit of an offset. So when you take the force out, you have a hyperpolarization phenomenon, and you have this kind of profile of the transduction current. Now, if you do the opposite, so you apply the force in the opposite direction, you don't have symmetry at all. Actually, you have the opposite of what you had before. Meaning, you push this way, what's going to happen is that the tip links are getting compressed. So there's going to be hyperpolarization, as you can see here. And then at the offset of the force, the molecular motors will start acting, and you will get a force like this. Now, a current like this. Now, if you compare this with this, clearly there's no symmetry at all. Actually, there's almost anti-symmetry. So how is it possible that in the case of C. elegans, you push and you get a current, you relieve the pressure, and you get a current which is essentially indistinguishable? So that's the problem, and that's how I got caught up into this. That's right. So this is the data that I showed here. This one. So they are applying pressure, then they keep the pressure for a while, and then they take pressure out. And this is what they measure. So worm by worm, you can have some differences, but when you accumulate all this data, these two peaks, they are essentially statistically indistinguishable. So there's some decent symmetry that, yes? Wait. Yes.
When you apply the pressure to these worms, is there any propagation where the actual position of the neuron is changing?
Right. So what's going on within the worm? It's not been quite done. Yes, certainly they move. So the neuron and the whole epidermis is moving. Characterizing them, it's something that we are doing, but clearly what happens is that things are rearranging within the epidermis. Yes. Okay, so that's the problem. And so how can you get symmetry? And getting back to Pacini corpuscles, they have this property. Pacini corpuscles, they already have this property of symmetry. This is a beautiful paper done by Lowenstein and Mendelssohn in '64. For reasons that I will tell you in a second, it's not useful for what I'll tell you in the sequel, because this model doesn't apply to C. elegans. But let me describe briefly, because it's a nice work. So let me tell you what they did. The reason why Pacini corpuscles attracted a lot of attention is that they are very visible. They have about the size of 1 millimeter, as you can see in this plot here. This is a Pacini corpuscle. It's about 1 millimeter in size. And the other thing which is very striking is that you have all these lamellar structures that you see here. So the interior of the corpuscle, it's made of a nerve, which is at the center here, and then it's made of a lot of lamellae that fill up the body of about 1 millimeter, that are connected by some filamentous, by some collagen and some other structural proteins that connect them elastically between each other. And then all this is in a soup of or collagenous substance, which is about 90–95% made of water. So you should imagine these lamellae connected, and then all this into a aqueous water solution. What's the function? Right. So what's the function of all this? What they did is this beautiful experiment. So they took a Pacini cell from cats, which is shown here, and then they put this under two plates, a plate which is movable, which is G1, and the plate which is fixed, which is G2. And what they did then, they have some light source, which is here, and then they have a photo detector here where they can measure the movement of the plate. And the experiment they did, in the wild type, if you don't do anything to the Pacini corpuscle, this is the response that you have. You apply pressure, you get a response. Then you have perfect adaptation on time scales of 10 seconds. And then you have again, at the offset, you get the response. What they did is that they removed one after the other all these lamellae. And they looked at what happens when you take off these lamellae. And when you take off these lamellae, this is the indentation, and this is the response. So you can clearly see that the role of these lamellae is to create some mechanical pressure inside the body of the neuron, and create the excitation, which is then producing the response of the nerve. So this is clearly showing that you lose adaptation as you remove these lamellae. And then they made a model where they model these lamellae by some elastic connections, and then they have some dashpot. So Lowenstein is the same person who did the experiment, and Schkalak, as you can tell from this graph here, is an engineer. So you make a model like this, and then the symmetry is coming from the fact that when you compress the corpuscle, you generate waves, pressure waves inside the neuron. These pressure waves, they propagate to the nerve, to the central nerve, and the central nerve responds to this pressure in homogeneities, and you have the response over there. Now, when you expand, you're going to generate again waves that are out of phase and rotated with respect to the previous ones, but you can still generate waves, and this is the way you get the response at the offset. So people were very happy. It's a nice paper. People were very happy with this explanation. It's 66, as you can tell. The problem is that then they started looking at other mechanoreceptor, touch receptors, and they realized that this explanation, even though it's so nice, it's actually very specific to Pacini corpuscles. The reason being that this structure, these lamellae, are unique to Pacini corpuscles. All the other receptors, they have this property of symmetry, but they don't have all these lamellae. So the problem is still there. Pacini corpuscles, there's a nice explanation, but it doesn't apply to all the other receptors. Okay, so let me tell you what we proposed and the evidence that we have in favor of a very different explanation that doesn't rely at all on the lamellae. And it actually relies on a generic property of all these receptors. All these receptors, they are located in the external part of the skin. So they are located in thin layers of the epidermis. And what we want to propose is that this property of symmetry, in this case, reflects the lamellae, but in general, it reflects the fact that these receptors are located in thin layers of the epidermis. Let me show you how the argument goes. And in order to do this, let's go to elasticity. And doing this slide, I realized how foolish it is to try to compete with Landau-Lifshitz, because I was trying to explain this with my own words, and I realized that I was doing so much worse than Landau-Lifshitz that I ended up copying Landau-Lifshitz and just putting it here. So you can read by yourself. If you have a thin layer of a material, of an elastic material, There's two contributions when you do elasticity. There's a bending contribution, and there's a stretching, compression, and stretching contribution. So let's get started with the bending contribution, which is shown here. The crucial remark, which is valid for thin material, first of all, I will be doing a plate. Of course, a cylinder is not quite a plate, but the same remark will hold for a cylinder. If you have a thin material, as you can read here, the forces in a thin material are very tiny. So the boundary condition at the top of the plate and at the bottom of the plate is that the z component of the force, so perpendicular to the plate itself, the boundary condition is that the force is 0. It is 0 in the upper layer, it is 0 in the lower layer, and since the material is thin, it's going to be essentially 0 throughout the whole shell. So what happens in a thin shell is that you have a stress tensor which is essentially in plane. All the forces are located in plane in the shell. So it's quite a special geometry, and this is written here. So the component sigma is the stress tensor. All the components that have z are equal to 0, are essentially 0, and the tensor is symmetric. So sigma zx and zy and zz is equal to 0 as well. So this is the basis for calculating the elastic properties of a plate. And the bending properties, in the bending, there's a so-called neutral surface. So when you do bending, when you just bend a surface, there's a so-called neutral surface, which is in the middle of the plate here, which stays isometric. So it doesn't get deformed. There's no stretching and no compression. The upper part is getting compressed, the lower part is getting compressed. So, and there's a variation as z. So if you calculate and put up everything together, you get E, which is the Young modulus of the material, and then you get H cubed, where H is the thickness of the material. Now, you can consider the stretching and compression, which is here. In this case, they don't vary over z. They are uniform throughout. So this is not an isometric deformation anymore. You get stretching or compression. And in this case, again, the same remark holds, which is the stress is in plane. So the only components that appear are in plane, xx, xy, and yy. xz, yz, and zz, they don't appear. In this case, because this is uniform throughout the thickness of the plate, you get, again, the Young modulus, and then you get something which is proportional to the thickness. Now, these two elements, they get combined together in the so-called Foppel-von Karman plate equation. It's an equation that combines these two terms here. And so the cartoon version that all this is solid physics. The cartoon version that we have for C. elegans in the paper is this one, where you indent, you're going to get a force which is going to be in a certain direction. Now, when you relieve the force, you're going to have, again, in plane. So you're going to have a force that in one case is going in this direction, in the other case is going in this direction, but when you move up and down, the forces have a symmetry that in one case is going this way, and in the other case is going in the opposite direction. Okay? So this suggests a gating mechanism, which is in cartoons, is the following one. So you remember this Hudspeth model of the trapdoor, which is going up and down, which is getting raised by the link. Now, in our case, we have the symmetry of the force which is swinging in the opposite direction. When you apply, it goes one way. When you relieve, it goes the other way. So a simple mechanism that can lead to the symmetry in the response that we have observed is to assume that there's, again, a trapdoor, but rather than having a hinged trapdoor, like in the case of Hudspeth model for hair cells, you have a sliding trapdoor. So when the force is getting applied, the trapdoor is going to swing in one direction. Then the friction with the surrounding medium is going to provide some relaxation mechanism which is going to bring the trapdoor back to the initial position and close the channel. This is going to provide, exactly like in Hudspeth model, the adaptation. This is going to close, and now when I relieve the force, the trapdoor is going to swing in the opposite direction, but it doesn't really matter for closing or not closing, whether it swings one way or the other way, this doesn't matter. The channel is open, and therefore you're going to have the current flowing. And so you get again the friction, and you close again this channel. Okay, so that's the cartoon. In order to convince, of course, you have to do a little bit more. And so bear with me for a couple of slides. I think it's interesting to tell you what we put in the model, which is very simple. So the equations. We suppose that there's an elastic component of the force. So when the anchor, which is this object, but we don't know exactly what it is, let's call it anchor. When it moves far away from the channel, there's an elastic contribution that tends to restore the distance between the channel and the anchor. So it's going to be brought back. Then we suppose that when the anchor is moving through the medium, we don't know what this medium is. It could be the cytoplasm, it could be the membrane, it could be the extracellular matrix. At this stage, we don't know what it is, but we just suppose that there's going to be some friction term between the movement of the anchor and the surrounding medium. So x prime is the point where the anchor is located at a particular time t. And then we suppose that when you apply a force, there will be a differential displacement between these different components. So the channels, getting back to your question, the channel is going to displace with respect to the extracellular matrix. The anchor is going to get displaced because of the application of all these forces. At this stage, I cannot tell you exactly how they move because it's not been visualized, but that's what we are assuming, that there's some differential displacement because of the in-plane forces that you have. And this will produce a displacement which is proportional to indentation. Indentation at the location of the ball that you produce. So there will be a profile of deformation. The profile of deformation will extend laterally, but it's going to be proportional to the amount that you indent with the ball at the location of the ball. So now with these three elements, it's a two-line combination of all this stuff. You put to zero the sum of the forces. So you apply the overdamped approximation, which in the molecular world is certainly appropriate because there's no inertia at the size of these molecules. And you get an equation which is this one. So it's dx dt plus 1 over tau, which is a combination of gamma over kappa. So there's a relaxation term, and then you have a forcing which is proportional to this chi, which is a constant that depends on the position, and then d zeta dt. So the indentation, d zeta dt, is the forcing of this element here. So this equation is very simple, but it already contains the two elements that we want, at least one element. If d zeta dt, zeta is kept constant, this term will disappear, therefore x will relax to 0, and you get adaptation. So the anchor is going to get back to the position of the channel, which is adaptation that we were looking for. Then, if we take d zeta dt, we go up and down. D zeta dt is getting reversed. So x has the same dynamics, just with a change of sign. Okay? But as I said, it doesn't really matter. It just means that the anchor is swinging in the two opposite directions. But now, and then we can... then the rest is, this one is standard thing. So these channels have been characterized. They have at least two states, a closed state and an open state. Actually, they have more. They have transient states as well, but it's not important. So we just assume that the free energy, that they obey detail balance. And the free energy difference is a constant term that makes the difference between the two. And then the stretching of the filament, so if the filament is swinging one way or the other way, it's putting tension, and it tends to open the channel and favor this state over this state when the filament is under tension. The only thing left before I can show you the results, is how to treat the different channels, okay? Because you're applying with the ball, you're applying a force here. Clearly, all these channels are not equivalent because they're positioned at different distances from the indentation point of the ball. So clearly, they will not respond the same way. We are doing something a little bit more precise, but the first thing which works pretty well is just to try something, is cheating, as we usually do with mean field, and just say the following thing. We treat all the channels that are close enough to the indentation point as one channel, as all the same, and then those that are too far, we put them to 0. They don't respond. So we just say that there's a radius of channels that respond all the same, and then those that are outside a certain radius, we just discard and we say they don't respond. So we replace the indentation profile by a Heaviside function. It's all the same, and then we put it to 0. Of course, it's an approximation, but it's, as you will see, it doesn't work badly, and it's a very good starting point. By the way, so we put this as an extra term, which is here. So when you look at, when you put this at the exponential, this term becomes x to the p. So, and the reason why we picked the power law is that there's evidence that when you indent a sphere or a cylinder, whether it's kept under pressure or not under pressure, the indentation field will be proportional to a power law of the indentation. So if you look at the deformation profile, it's going to extend laterally proportional to the power law of the indentation. That's a known result. You can look in this paper here. Okay, so these are the elements. We have an equation for x, which is this one. Then we have the channels that obey this equation here, and we can just apply the indentation and predict what the current is going to be and compare with the experiments. So these are the results for the step. We predict the mean current, which is shown in black, and then we give an estimate of what the standard deviation is going to be based on fluctuations and the number of channels. So this is a step in indentation. This is the response of the system. Green is experimental data, and black is what we predict here for this tree. You can also look at the probabilities here for the channel to be closed or to be open. And as you can see, when the force is being applied, this one in blue is the closed state. It goes down. There's a probability. This is a transient state, and this is an open state. So you can see there's a higher probability for the channel to open. And then when you relieve the force, then there's adaptation, which comes here. And then you have, again, a response when you go when you take the force out. This is one set of data for steps. They can deliver all sorts of stimuli. So, for instance, they can deliver sinusoids, as you can see here. And you won't be surprised because of adaptation. If the frequency is too low, so it's too slow, the system will just adapt, will just keep following, and there's essentially no response, as you can see here. Well, the frequency increases, you start getting more of a response, and then when you get into the 10, 30, or 100 hertz, you get more of a response. Now, when I saw this plot first, I got surprised because you can see one thing, which is that if the indentation has a certain frequency, the response has a double frequency. So I thought first as some quadratic normal form, but then I realized that it's just a symmetry because it responds to the onset and the offset. It's responding actually at this and at this the same way, and therefore you get double frequency. It's just the symmetry between onset and offset of the response that you have. I'm running out of time, so you can predict that this. So for reasons that have to do with the technique of the cantilever, it's not possible to deliver at this stage, but they are constructing a different system of delivery that will allow to deliver stimuli in the several hundred hertz. At the moment, we are limited to stimuli, to frequencies that are maximum in the range of 100 hertz. So in the 100 hertz, what you can see here is that the system is not able to respond anymore. The reasons is that there's inertia, there's a switching time for the channels to go from open to closed. So there's an intrinsic time which is of the order of 10, 20 milliseconds. Therefore, if you start going too fast, the system is not able to follow anymore these frequencies. There's some inertia at the level of the channels, and you see this flattening out of the response. We can predict that the system is going to act as a bandpass filter. I won't give you details, just this. So this is our prediction for the bandpass filter, and it's not tested yet, but that's a prediction that we have. And then let me show you ramps. So as you would expect, when you have a ramp, so you fix the initial level and you fix the final level of the indentation, but you go from one to the other with different speeds. And again, if you go too slowly, the system adapts and doesn't respond, while the faster you go, the stronger is the response of your system. And you can predict how, so these are the experiments that are done. F is the force and Z is the indentation. And indeed, when the faster you go, the more you get of a response. And these are the experimental data, and this is what we predict. So there's again, a good, very good agreement. And the data that I like the best is this one, that of course my collaborators wanted to discard, because what happened is the following thing. They were trying to generate a ramp, as you can see here, but the feedback system didn't quite work. So there was a slippery of the system here, as you can see. And what happened is that the feedback control was not able to control, and there was a mismatch in the feedback system, which produced this crazy behavior of the indentation that you can see here, with all these fluctuations, these tiny fluctuations that you have here, due to the fact that the system was trying to implement the feedback control. And this is the response of the current that clearly is very regular, but still with our model and all these control parameters that we fitted with other things, we can just reproduce this behavior. So that's something that I was quite happy with, because we couldn't fit parameters anymore, and this is really a complicated behavior that we could reproduce. Okay, so I'm really running out of time, so let me just conclude by saying what we are doing right now. What we are doing right now is try to go beyond mean field, and the way to go beyond mean field is to do what we should do, which is look at the behavior of a cylinder that gets indented with a ball here, and looking at the profile of deformation. Now, the equation for a cylinder that gets deformed, the thin cylinder that gets deformed, are written here. I will not explain this equation. This is the bending term, and this is the stretching term that you've seen before. These are partial differential equations. Fourth order, the typical elasticity. I just want to give you the physics which is in there. The physics is that the strain in the system is still small. Okay? So there's still a Euclidean relation between stress and strain, but there are, as you can tell here, as you can see here, there are quadratic nonlinearities. These quadratic nonlinearities are what is called in elasticity geometric nonlinearities. They have to do with the fact that when you deform a cylinder or a plate, you can have very strong deformations that have to do with the fact that the cylinder can be deformed without stretching or compressing. So this generates strong nonlinearities that have nothing to do with the response of the system. The system is still Euclidean, but there are nonlinearities because deformations are strong. And this is what is included up there. Okay? So doing this, we can look at the profile and get a little bit closer to what you were asking. So the body of the worm can be stained with GFP, and then you can start having a sense of how it's getting deformed, at least the epidermis. And this is the profile of the experimental data of the profile of deformation, which is well captured by this kind of models if you take a pressure which is in the order of kilopascal, tens of kilopascal. And then the relation between force and indentation is well reproduced if you take a Young modulus, which is in the order of megapascal. Okay? And then you can again look at the profile, how it extends as a function of the indentation, we get indeed the power law, which is reassuring. So that's the thing that we are doing at the moment. And the reason why this is going to be important is, first of all, to confirm what we have done by mean field before, but most important because we want to do two things. One is to look at the case where you put multiple points of stimulation. So you have, for instance, two stimulations, which is the case of the worm. It's not stimulated at a single point, it's stimulated at multiple points. And the other thing is that we want to study the noise, because we were predicting mean levels of the current, and we want to study the level of noise. And with this, I'm done. And these are people I've been collaborating on this project. Amy Eastwood and Miriam Goodman, they are at Stanford Medicine. Beth, Sung Jin Park, and Brian, they are at Stanford Mechanical Engineering. And Alessandro, despite his shirt, he's Italian, he worked with me. And he was paid by Milano University and is finishing his PhD in the next 3 or 4 months. That's all. Thank you.